Today I'm going to show you a new service that we have in PATRIC called codon trees. It's a new service that we have to build phylogenetic trees, and it takes both protein and amino acid sequences and combines them together to give a pretty powerful tree. Let's go into services and find phylogenetic tree. So click on that. And this opens up the interface for the phylogenetic tree building service. Tutorials for that service and additional information can be found by clicking on these icons here at the top. We have two phylogenetic tree building methods, codon tree and all shared proteins. All shared proteins is a blast analysis that takes a very long time. So we're in the process of replacing it with codon trees. So the codon tree algorithm looks for shared protein families across all the genomes in the selection. And once it looks for protein families, the PATRIC's global families that are shared across all the genomes, and it looks specifically for protein families that all the genomes have only one copy of that gene within them. And when it finds that, it gets both the nucleotide sequence of the gene and the amino acid sequence of the protein. Now, here you can have input genomes, add at least 4 or up to 100. Actually, I've built a tree with 200, so you can go higher, but always keep in mind that the more genomes you add, the longer it's going to take. And also, the more genomes that you add, especially if you're getting ready for a publication, even 100 genomes is really hard to resolve in a figure. So think about what it's going to look like in the end for just general knowledge and experimentation to try to see which groups belong together, how they settle out, and then choose specific genomes from that. Go ahead and load up on the genomes if you don't mind waiting for the time. But for your publications, you might want to be a bit more careful in what you choose to always have that in mind of what's going to look good in the publication. You can add select genomes to a group, and to do this, you could just start typing. Like I could type Yersinia, and it's giving me a bunch of choices. And PATRIC has thousands of Yersinia genomes in here. And so you need to know something about this gene, the genomes that you want to add here. Generally, you want to add your private genomes through this or specific groups. So if you click on this folder funnel icon here, I can find specific groups of genomes that I'm interested in adding. For instance, let's say I click off my genomes, click off all other public genomes, and click on reference genomes. And then I have to get rid of this guy here. And I start typing, and it's showing me here all the reference genomes that there are within that group, which I use the genus name Yersinia. So I'll just pick one of those, click on it. And now I have to add it into this selected input genome table. To do that, I click on the Add button. In different tutorials that we have created for PATRIC, I show you how to create genome groups. You can also add a genome group to a tree, which is a lot easier than adding 100 genomes individually. Trust me. So you could go directly to your workspace by adding the folder, or you can click on the down arrow, which will show you the genome groups that you created most recently, or you can start typing and it's going in to look at genome groups that match your name. So plasmids, that sounds interesting, so I'll click on that. And then I need to add it to the group. And I want additional groups. So Yersinia, I'll do these reference genomes. That looks interesting for my tree. And add that to the group. And you can see it's showing them in the order that they were added. And I want to add one more. And I'll add the group called Yersinia group for tree that I added earlier, and add that to the group. So I've got all these genomes I want to add to the group. Let me point out that you can mouse over that to see the genome ID, which is the unique identifier. But let's say there's, oh no, I made a mistake. I don't want this genome in my tree. It's easy enough to remove it just by clicking the X here, and the genome disappears. So instead, we're going to build a tree with this group. Okay? We need to have a folder for the group. So you can create a new folder by clicking on the output folder here, and it's going to show what's in my workspace, which is very messy. So I want to create a new folder because I'm trying to clean up my app. Yersinia trees. Let me see. I want to call that folder this. Okay. So now I go back to this home folder. Well, if I click on the down arrow, it shows me what I just created. Oh, it looks like I've used that before, but I'll click one of them. So it's going into the Yersinia trees folder. And I need a name for my genome, so I'll call it mixed Yersinia genomes with plasmids, because remember I included plasmids with this. And the reason I'm showing you this particular example is in another tutorial that we've done showing you how to determine if you can build, if the genomes you've selected will generate a tree. We warned against using plasmids, but I'm going to go ahead and create a tree with plasmids just so you can see what it looks like. Now, the number of genes, if you click on this down arrow, you can see that we can use 10, 20, 50, 100, 500, or 1,000 genes. The more closely related your genomes are, in order to get clear resolution, the more genes you will need to add. But when you're toying with things and just trying to figure out where things are, if you choose a small number of genes, it'll run a lot more quickly. So that's what I encourage you to do. If things look good with 10, go ahead and choose those. Another thing to keep in mind is that when you are submitting your tree as part of your study for publication, some reviewers, if you only use 10 genes, some reviewers may not think that your tree is strong enough to determine the evolutionary phylogeny of your group. So for a publication, I would recommend you would do 100 or more. But for this one, we're just messing around. We want to see what we've got, so let's choose 10. Now there are two other things that you can select. This one is maximum allowed deletions, 0 to 10. What this is saying is when PATRIC builds the tree, and using codon trees in this algorithm, I told you earlier that it looks for protein families, the PG fams that we created in PATRIC, that all of the genomes in the group share, and they only have one copy. Well, sometimes you don't have a very good genome, and it may not have all the proteins that the other ones have, but you still, especially if it's your private genome, you want to try to build a tree with this. So you can say, I allow for 1, 2, 3, 4, all the way up to 10 of the genomes in the protein families in my group to not be complete. That means that all the genomes in the selection don't have a copy of this. You can also, with this maximum allowed duplications, it's looking for, it allows you to use protein families that might have one or two or three or more copies of an individual gene. This happens sometimes. There are duplication events or other genes have entered the genome via plasmids, and sometimes they have a couple copies of that. So this will allow you once again to choose the 0 to 10 number. You may ask, well, which version of the gene does it use then when it's building the tree? What it does is it takes the gene that has the strongest homology to everybody else in the group, and it chooses that. So here we have the genes. I'm not going to choose any of these for this, but we can submit the tree. When this turns blue, you can submit it. So let's go ahead and submit the tree and see what happens. And it gives me a message that tells me that this job has been submitted. It can take hours to a day to complete, depending on the number of genomes and their size. What I want to do really quickly is change this now, because remember, we are including a genome with plasmids or a tree with plasmids. I want to submit a new job without those plasmids. So I'm going to go up here and delete that. And now it's just these genomes without the plasmid. And I'm going to call this if I could spell, Yersinia genomes 2019. And let's do the 10, 0, 0. So, another thing you can do is submit. This makes it very easy to submit successive jobs of the tree, just doing that kind of function. So let's submit that. Okay. And once again, it gives me a message to see that I have submitted this job. So how do I find these jobs that I've just submitted? I can click here down in the lower right, this jobs box, and that's going to open up my jobs page. And oh, look, the mixed Yersinia genomes with plasmids is already completed. I'm very excited to see this. This job has been completed. So to see it, I click on this, and the vertical green bar tells me that I can view it or report an issue. When you have a job that's failed, which I occasionally do, well, I don't see any right here, but I can click on the report issue, and then I can report the problem that I have with that. Or let's say you identify some other problem with the tree. You would need to provide some text about what actually you're complaining about, and then you could submit the job. But I want to view my tree because I didn't expect this to complete, right? So let's click view. Oh, all I see is this HTML. And I don't see things that I would expect to see, like a NUIC file or things that belong with trees. So let's click on this. And here I can view it. And look what it says. So here's the tree report. It tells me that I chose 62 genomes. I didn't have any deletions or duplications. I requested 10 genes. Oh no, it didn't find any shared genes, and it didn't find any shared amino acids. If I scroll down, the tree building algorithm we use is RAXML, and the report tells me RAXML didn't even run. It couldn't even generate an alignment. So I look here, and here are the genome IDs and the names of the genomes. This tells me the total number of genes and how many genes they used, and none of them, because I couldn't build a tree with that. But one thing it does tell me, and this is my favorite part of this, even for trees that succeeded, this will give you hints on how you can improve the tree. So if I go here, it tells me if I delete these genomes, and who can remember what all these numbers mean, but I can copy it and then do a find. then I can see if I delete that from when I'm trying to build this tree, if I delete this genome, and in fact, I would need to delete all of these genomes, then I would be able to increase the number of genes that I could see. So that's, that's kind of a downer report. I didn't get to see the tree. This is when people go in and they generally give me a message and say, I don't understand what happened, why my tree didn't build. That's when I tell them to watch the other webinar we have on Are My Genomes Treeable? So let's look at a tree job that completed so you can see what that looks like. So let's click on jobs again. And let's see, I can go up here and I can go into all services and I can pick codon tree to see genomes, trees of mine that completed recently. Let's just click this one, or let's click this guy. This guy looks good. Okay, and let me get some more space here. So this, when I see these kinds of folders, it indicates to me that I have a tree that succeeded that I can be very excited about. So this is what the HTML report. So first let's look at that and let's view it. And I opened that more so I could see the real estate. And here's the tree. And look at the support values of that tree. They're great so far. That one's kind of weak, but oh, look, supporting these different clades, the bootstrap support that looks at the differences between the two clades is really well supported. So I'm happy with this tree. It looks like a pretty good tree. And because I can see this here, this tells me that this tree has been midpoint rooted. When I go into the tree statistics, it tells me the number of genomes. It tells me what I selected, which was 0 for deletions and duplications. I originally wanted 20 genes. It found 20 genes. It found 20 proteins. And then it tells me the number of aligned amino acids and the number of aligned nucleotides. When I'm writing a paper, I often bring these numbers in. And it's going to be hard for reviewers to argue with really strong numbers here. Next, it tells me the RAXML command line. And then it goes through the genome statistics, what the genomes were. It looks like with this tree, I requested the 20. And there are a lot of genomes here. This is another good thing. I would download this thing and provided as supplementary material. Often a reviewer will ask, well, what genes did you use to build the tree? Well, here they all are. So that's for them, easy for them to see. And you can see that because I asked for 20 genes, I got 20 genes. It doesn't show me any suggestions for improving this tree because I got everything that I asked for. Let's go back up here into the breadcrumb and click up one to go back to the job landing page. And I just want to show you the other things that are provided with when this algorithm runs. You can get a PDF of the tree, a PNG of the tree, and if you want to view it, you can, of course, view it. Oops, there we go. An SVG, a scaled vector graph that should be publishable. But this is one that I often use are the Newick files because sometimes the trees that we have in PATRIC, like here I've highlighted the Newick file and I can click on the tree view here. You can see what it looks like in PATRIC. So this is the same tree. One thing that's good about this particular view is, let's say I wanted to do a subtree on this group. I ran a whole bunch of different genomes together. I didn't realize they belonged together. you can click on that and you notice it checks all of those genomes. And then I could actually group those and run a new tree on that. But the NUIC file that we talked about earlier is the instructions for building a tree. You can open this in a program like Figtree or the Interactive Tree of Life, and you can rearrange the tree. You can't change the branching patterns themselves, but you can flip them and arrange them in a way that that will make for a more attractive tree. And also you can annotate it with metadata that you often see. In fact, in another webinar, we'll show you how to do that. This tree building algorithm also has a huge number of files. If you want to see even more, you can click on the detail files and you can see, this is like a computer scientist's dream. It'll give you all the statistics on everything. And actually, I get questions about users want alignment statistics or the alignment, the PGFAMs and genes included in the alignment. This would be something I would take and just use a supplementary material in a paper. All of that is here. Now, before we leave this webinar, I want to show you, let's go into Help and go to the tutorials. And we need to click on Genomics. And then we need to find Building Phylogenetic Trees, codon trees. So let's click on that. This will show you absolutely everything that I just stepped through in this short tutorial. But what it also gives you, and this, I've tried to build these things so that you can use this in your publication, because what I want it to be easy for you to run the analysis and then just know exactly how the tree was run. You could put something like this here into your publication. So it tells you that the protein and gene nucleotide sequence were selected, genes using PGFAMs. I reference PGFAMs above. The protein sequences were aligned using muscle, and the nucleotide coding gene sequences were aligned using the codon align function of BioPython. A concatenated algorithm of proteins and nucleotides were written to a phylet formatted file, and the partitions file for RaxML was generated. At the very bottom of this, which I'll scroll down, I don't want to give anybody a headache, but I provide all the references because we want you to use PATRIC, and we want it to be easy for you to use PATRIC. So the references for all of the things that are used in this algorithm are included here. So you have the tree building pipeline that you can use, codon trees, the descriptions that you can put directly into your publication. But please remember to cite PATRIC too, because it helps us if you cite our resource. Thank you.